This is part 2 of chapter 15, the mechanism of pathogenicity. As we started in part 1, we're going through the process by which a microbe will cause disease in an individual. And to begin this process, we would start with a person who has a predisposing factor and is exposed to the microbes. The microbes then would enter by their particular portal of entry in sufficient numbers to cause infection, and they're able to adhere to the tissues. Once they get into the individual's body, then they have to evade that person's defenses. All of that I've talked about before. For this lecture, we're going to start with how those microbes then actually cause damage to the host cells, and then the portal of exit they use to leave the body to infect others. Once the microbe is inside the person's body, there are 3 basic ways that they cause damage to that individual's body to create the disease. The first way they can cause damage is by direct damage, directly damaging tissues. The second way is by taking nutrients from the host. So by taking nutrients, they're depriving the host tissues of nutrients, and that's what can cause the damage. And then the third thing is toxin production. One type of damage that a microbe can do to the human is direct damage. So direct damage is where the microbe is actually directly damaging cells. This can be done when the microbe enters the body. An example of this is Nectator americanus. Nectator americanus is the American hookworm. And if you remember when I talked about the life cycle, what the hookworm does is it attaches to naked skin, and then it can burrow through intact skin to gain access to the circulatory system. So this is obviously direct damage that the hookworm larva causes when it enters the human's body. Another type of direct damage can be during viral replication. So if you have a virus that lyses cells, like a naked virus, then the cells that it infects will be lysed, and that will damage the tissue. So those are 2 types of direct damage where the microbe is actively damaging cells it comes in contact with. The second type of damage that microbes can cause in a host is by nutrient uptake. They're utilizing the nutrients for their own metabolism, and then that deprives the host of the essential nutrients. Viruses are the only type of microbe that would not cause this type of damage because viruses don't metabolize on their own, so they would not take nutrients away from the host. When we think of this kind of damage, we usually think of parasites. So the parasitic worms or other types of parasites. This is a tapeworm, which is a cestode, and remember it has the scolex down here. That's the scolex, the head. That has the hooks and the suckers, and extending out from the scolex are the proglottids all the way to the end. At the end of the tapeworm, you have the mature proglottid here that is full of fertilized eggs. So that's just a review of the structure. But tapeworms, the worms, they are parasites living in the intestines, absorbing nutrients from the host, and that's how they cause most of their damage. Bacteria can also cause damage by taking nutrients from the host, and the most common way they do this is by lysing red blood cells. So these are the red blood cells, and they do this to release the iron. So bacteria have a high need for iron, high requirement for iron. So often bacteria, if they get access to the blood, they will lyse the red blood cells to take hold of the iron, and that causes anemia. Now, viruses cannot take up nutrients from the host, but they do cause cytoplasmic effects. And again, this is a review from when I talked about the viruses. Viruses, when they infect cells, they can cause changes in cell structure, and that can affect how the cells function. Two types of cytoplasmic effects are the multinucleate cells. Multinucleate cells, that's where you have several cells that are infected by a virus and they fuse together to form a multinucleated cell, so a cell with multinuclei, or sometimes called giant cells. And so this is an example of a giant cell that I showed you before. So a lot of cells are fused together, multinucleate cells. And also I talked about inclusions. So inclusions you can see inside the cells. These are darker staining areas that are not found in normal cells. And these are sites where the virus is replicating. It can be copies of the genome. It can be capsids are building up in that area, capsomeres or capsids, or the mature viruses. But when you see these inclusions, that often means that the cells can't function normally, and so that would be having an impact on the host. This is just a table from your book that gives you the name of different viruses and the different type of cytopathic effects that they cause. So some of them can cause actual cell death. A lot of the others have different types of inclusions that they produce inside the cells, and the measles virus, this causes those giant cell formations to occur. The third way that microbes can damage hosts is by releasing toxins. There are two basic types of toxins, endotoxins and exotoxins. And the difference between the two is that endotoxins are part of the bacterial cell wall and are released at death. Exotoxins are made inside the cell and secreted out. So you need a living cell that can create the exotoxins and secrete them out. The next couple slides, I'm going to compare and contrast endotoxins and exotoxins. So even though their names are very similar, they are very different in structure and have very different effects on the host. The first type of toxin I want to talk about are the endotoxins, and they're basically just one type, and that is lipid A. And if you remember, I talked about lipid A before. Lipid A is on the gram-negative bacteria. It is a molecule in the outer membrane. So remember when I talked about the outer membrane, there is the lipopolysaccharide molecule, and when the cell dies, it is split into the O polysaccharide and the lipid A. So the O polysaccharide was used for identification of different species, and the lipid A is the endotoxin. The first toxin I want to talk about are endotoxins. So I'm going to list out some characteristics of the endotoxins, and you will use this to compare to exotoxins. Some of these things I've already talked about, but just to put it all together, endotoxins are associated with gram-negative cells because they are part of the outer membrane, specifically part of that lipopolysaccharide molecule. Because they are a normal part of the structure of gram-negative cells, the gene that encodes them is on the chromosome because it's a normal part of the structure of the cell. Next, I mentioned they are released at death. Again, they're part of the normal structure, so the only way they get released from the cell is when the bacterial cell dies and breaks down. An important aspect of the endotoxin is that it is a lipid. So everything you have learned about lipids applies to the endotoxins, and the most specific thing is that they are non-polar, they are hydrophobic, they do not interact with water, which means they have very poor diffusion. And because of that poor diffusion, they do not diffuse well in blood, so it takes relatively high concentrations of them to have a negative effect on the person. And the effects that they have on the person are usually very general and systemic. So usually endotoxins will cause fever, which is a systemic reaction, and they can also, if you have high enough levels, cause shock. Because they are lipids, you cannot inactivate lipid A or endotoxins. Heating them doesn't denature lipids, so that will not inactivate them. And also because they are lipids, your body will not produce antibodies to them. So even if you are exposed to the endotoxins, you will not produce antibodies, which means there's no way to vaccinate for them, because vaccines rely on the idea that our bodies produce antibodies. So if we cannot produce antibodies to the lipid A, then there's no possibility for a vaccine. The second type of toxin are the exotoxins, and I will go through the characteristics for these. And again, you need to be able to compare and contrast to the endotoxins. So exotoxins. Exotoxins are not associated with any particular type of bacteria, so they can be found on gram-negatives and gram-positive bacteria. These genes are often found on plasmids. So again, back at the beginning of the semester when I talked about plasmids, I mentioned that they have antibiotic resistance genes and toxin genes. So that's where you find most of the genes that encode for the exotoxins. Also mentioned before, exotoxins have to be produced and secreted by living cells. So if you kill the cell, then that cell can no longer produce exotoxins. In terms of structure, exotoxins are proteins. So proteins, and that's very important to the rest of their characteristics and their function. So again, when you hear about proteins, two things that should pop out right away is that three-dimensional structure is really important to their function, and that they tend to be highly specific. When I talked about enzymes, that came up a lot with the enzymes. It comes up with the exotoxins. And when I talk about antibodies, which are also proteins, this will come up again. Because exotoxins are proteins, that means they can interact with water really well. So they can dissolve in water pretty well and diffuse through your blood. So that means that you only require very low concentrations to have the negative effect. And because these are proteins and proteins tend to be specific, these exotoxins have very specific symptoms. And there are hundreds of different types of exotoxins, and they all have very specific targets and effects on the tissues. Because they are proteins, you can inactivate them. Think about the enzymes, all the different things that could have an effect on the 3D structure of the proteins, the enzymes. Those are same things that can inactivate the exotoxins. So again, because these are proteins, their three-dimensional structure is important to their function. So if you denature them, you will inactivate them. So simple things like heating them and changing the pH can inactivate them. Also, because they are proteins, your body can make antibodies to them. So it is possible to have antibody production against an exotoxin, which also means, hypothetically, we can produce vaccines for them. So it is possible to produce vaccines for exotoxins. So while endotoxins are going to have very general systemic effects on the person, like fever and shock, exotoxins are going to be produced by bacteria, secreted out into the body. They will have target cells, specific target cells, and within those target cells, they're going to have very specific effects. While there are hundreds of exotoxins, they tend to fall into 3 different groups depending on their function, on their effect on the cells. First group are the AB toxins. Second are the membrane disrupting toxins. And third are the super antigens. This is a table from your book that goes through different diseases caused by exotoxins. It gives you the disease name, the bacterium, and then the type of exotoxin. So quite a few of these exotoxins are AB toxins, a couple are membrane disrupting toxins, and then super antigens. So the super antigens. I'm going to go through and talk about these 3 subtypes and basically explain what they do. The easiest to explain, of course, are the membrane disrupting exotoxins, and those, of course, rupture membranes. And often the target are the white blood cells. So this is a blood smear. These are the red blood cells, and these are the white blood cells. And this is the effect of an exotoxin. So disrupting the membrane, which will effectively kill the white blood cells. And of course, the benefit to the microbe is the fewer white blood cells are around, the fewer cells are attacking the microbe. The second type of exotoxin I want to talk about are the super antigens. And this will make more sense after we go through the immune system. But the word antigen basically means a molecule that stimulates the immune system. So what the super antigens do is they overstimulate the immune system. So to do this, you have the infection, the bacteria. The bacteria secrete the super antigen, and the super antigen stimulates the immune system to secrete cytokines. And cytokines are like signal molecules. The white blood cells use them to communicate with each other. But the super antigens are overstimulating the white blood cells, so they are producing too many cytokines, and sometimes this is called a cytokine storm. And there are so many cytokines that it overstimulates, overaffects the body, and it can cause all sorts of effects, and mainly what the result is shock. And shock, if it's not treated soon enough, it can cause death. So super antigens overstimulate the immune system, which results in shock. Toxic shock syndrome toxin is associated with toxic shock syndrome, which is associated with tampon use. It was an infection that was facilitated by super absorbent tampons, and it allowed a staphylococcus to overgrow, and that staphylococcus produced a toxin. And one of the effects of that toxin is scalded skin syndrome, where the skin starts to slough off. So in addition to causing shock and causing death. The last type of exotoxin I want to talk about are the AB exotoxins, and that's probably the majority of exotoxins are AB type, and that means that the exotoxin has 2 components. The A is the active component, and the B is the binding component of the exotoxin. So this shows a live bacterium that is translating the exotoxin protein. The exotoxin will be secreted out by the living cell. And then the binding component is important for recognizing the appropriate target cell. So the B part of the exotoxin recognizes the appropriate target cell, and then the A component, which is the active component, will have the negative effect on the target cell. And different exotoxins have different negative effects. But an AB exotoxin has 2 parts. The B component is used to recognize the specific target cell. The A component has the negative effect on the target cell. While there are hundreds of different types of exotoxins, there are just a few that I want to talk about. The first one is streptococcal pyrogenic exotoxin. So streptococcal, it is associated with Streptococcus pyogenes, and pyo refers to fever. So Streptococcus pyogenes causes strep throat, and it also causes scarlet fever. And the difference between strep throat and scarlet fever is whether the bacteria has the gene to produce this exotoxin. And what the exotoxin does is it damages the capillaries so that the capillaries in the skin will leak blood, causing this rash. So that's the scarlet fever aspect of this infection. Often starts in the face because the streptococcus enters through the respiratory system and then spreads to the rest of the body. The second type of exotoxin I want to talk about is cholera enterotoxin. Entero basically refers to the lower digestive system. So the cholera enterotoxin causes massive diarrhea, which is treated by rehydration. So that's the picture of some cholera patients. What the enterotoxin does is it causes the cells lining the large intestines to release electrolytes, and that causes the cells to release water, and that causes that very watery diarrhea. And the dehydration that can occur can kill an individual in hours if it is not treated. The next two exotoxins are two of my favorite exotoxins, and the first one is botulinum toxin, which is produced by Clostridium botulinum. And the target for the botulinum toxin is the neuromuscular junction. So as a review of the neuromuscular junction, here is the axon, so part of the neuron, and this is the muscle cell down here. And you should know that they do not touch. There is an empty space between them called a synapse. And what the neuron does is it releases neurotransmitters, acetylcholine, which will bind to receptors and stimulate a contraction in the muscle cell. Well, what botulinum toxin does is it prevents this from occurring. So it prevents the receptors on the muscle cell from being stimulated, so that prevents contraction. The effect on the patient is paralysis or hypotonia. They cannot contract their muscles, so they have sort of, if babies have this, a very floppy appearance. This is an actual baby who has botulism. For babies, one of the things you're told is you should not give a child under the age of 2 raw honey, because honey can sometimes contain Clostridium botulinum. And if you give that to a child under the age of 2, their immune system is not mature enough yet to kill the Clostridium off, and the Clostridium can survive in the baby's stomach and produce botulinum toxin, which would create botulism in the baby. The other toxin is tetanus toxin. Tetanus toxin is produced by Clostridium tetani, and tetanus toxin also has an effect on the neuromuscular junction, but instead of preventing contraction, what the tetanus toxin does is binds to the receptors on the muscle cell, stimulates them to contract, and then prevents relaxation. So the tetanus toxin causes the muscles to contract, and then they cannot relax. An older term for tetanus was lockjaw, because the muscles in their jaw would contract, and they were unable to open their mouths. An even older name for tetanus was back break fever, and they called it back break fever because they said only the patient's head and heels were able to remain on the bed because the muscles in the back contracted so strongly that they were arched that way. That's probably an exaggeration, but this is a photo of somebody who does have tetanus, so you have the same kind of thing happening. I find botulism and tetanus very interesting, the toxins, because both of them come from related species, Clostridial species. They both have an effect on the neuromuscular junction, but they have different effects because, again, they're proteins. Proteins are specific. Botulism toxin prevents contraction, tetanus toxin prevents relaxation. And both of these can be deadly because they can both have an effect on the diaphragm. So actually untreated within about 8 hours, either botulism or tetanus can kill a person because it will prevent the diaphragm from functioning normally. The last part of how a microbe causes disease in a person is the portal of exit, because the microbe is going to leave that individual to go infect others. And I've talked about the portal of exits before, and the most common ones are the mucous membranes, first in the respiratory system, then in the digestive system, then in the urogenital system, and also the conjunctiva. Conjunctiva is the least common of those mucous membranes. Then you also have the skin. So the skin is a portal of exit, and the very last one is the blood with insects or syringes. So, as a review for this chapter, in order to have a microbe cause disease in a person, the person probably has some predisposing factors. Then the microbe has to enter through its preferred portal of entry. You have to have sufficient numbers, meet that ID 50. Those microbes have to use different structures to adhere to the individual. Then the microbe has to evade that person's initial defenses. Once they have done all of that, then they are starting to cause damage to the person's tissues. And the last stage, of course, is to leave that individual to infect others.